Hello, I'm back. So we will continue with gene regulation. Now, when we talk about gene regulation, we're going to look at three different operons. One is the like operon. Like operon is an inducible system. It has repressor molecules and also has inducers. But at the same time, it is also governed by mechanism that allows it to shut off it when glucose is present, and that is called catabolite repression. In the tryptophan operon, there's also a repressive system. In tryptophan operon, it is a repressible system. That means it is on and it will switch off when it's not needed. So it's a repressible system. It has a repressor that is expressed as inactive, but can be activated if needed. Then it will switch it off. But there's also another mechanism which is called the attenuation. The attenuation depends on coupling of transcription and translation. As the transcription produces mRNA, ribosome starts translation. That region has 4 regions of homology, 1, 2, 3, and 4. If tryptophan is available, then ribosome proceeds as normal and cover regions 1 and 2, and then 3 is not. When 3 is produced, 2 is occupied, so 3 and 4 will bind. That will give you the termination sequence, the termination hairpin structure. But if there is no tryptophan, ribosome will be stalled at area 1. And because 1 is, it stops there because there's no tryptophan to be put in the tryptophan codons. 2 and 3 will bind, and because 2 and 3 will bind, you don't get the 3 and 4, and then that allows polymerase to continue transcription. Okay, so now we're going to look at the Arabinose. Okay, I will go forward. Okay, let's read here. Arabinose, what is special is the AraC protein. So Arabinose codes for 3 enzymes that are required to catalyze the Arabinose. This is sugar Arabinose. So you got Arabinose isomerase, ribokinase, and also ribulose 5-phosphate epimerase. So these are the BAD. All right, but the 3 structural genes here, you have the BAD. Are genes that are arranged in an operon, and that is regulated by the AraC. So AraC is the control protein, right? Now, there are 4 important regulatory sites. So these are regions of AraC binding. So you have the ARA01 here. This is an operator. AraC binds to the site and represses its own transcription from the PC promoter. PC means this promoter is to produce more C. If AraC binds to ARA01, it prevents from producing more C. In the presence of Arabinose, however, when AraC bound at this site helps to activate. So it has 2 functions. So if AraC binds at ARA01, it does 2 functions. It stops producing C, but it also promotes BAD expression. Right? So see that there are 2 functions. Now ARA02 is also an operator. AraC can bind here as well. If AraC binds at this site, now what it does is that if it binds there, it can also bind simultaneously at ARA1. So how can one protein bind at 2 sites? What happens is that if it binds at ARA02, it also binds at ARA01. So basically what it does, this is the figure. So AraC binding at ARA02, but at the same time it will bind at ARA-I. It makes the DNA loop. Okay, and when it's looped, it's like a physical blockage. It's looped. So how can polymerase function? All right, so that's why this operon is special. So depending on where C binds, and depending on whether Arabinose is present or absent. If again I'll repeat this. If there is presence of Arabinose, AraC binds better to O1, and because of that, it stops AraC and also promotes BAD. But in the absence of Arabinose, AraC binds ARA02 as well as ARA-I and makes a loop. Okay. Um, so that here talks about the AraI is an inducer site. AraC bind the site can stimulate binding to AraO2 site to repress transcription. Okay. In the presence of Arabinose, AraC is bound to help activate. So we've seen that already. If it binds there, you have the CRP site. CRP site is the same as the one you see in like operon. Okay. So here is the whole story about Arabinose. So when Arabinose is present, there is no need to express the structural gene. Sorry, when Arabinose is absent, when there's no Arabinose, so of course you don't need the gene. So AraC does blocking by binding simultaneously to AraI and AraI2. So this is what you get. It's like that, it's sort of folded. As a result, the intervening means the DNA in between is looped. These two events block the PBAC promoter. It blocks that promoter because it's bound there, so polymerase cannot bind. Of course, polymerase also cannot access because this thing has the region has looped. AraC also prevents its own expressions. Remember previously we saw that binding at ARO2 also stops production of itself. If you remember, allolactose is a positive feedback regulator. It produces more of itself, but in this case, it is a negative feedback. All right, prevents its own expression. Thus, is an auto-regulator with own action. This makes sense. There is no need to overexpress AraC. If the concentration falls too low, then traction of AraC resumes. When Arabinose is present, right? If Arabinose is present, then you need to start metabolizing Arabinose. AraC, remember this word, allosterically induces it to bind to AraI instead of AraO2. So AraC now doesn't like to bind with AraO2, it likes to bind to AraI. If glucose is also absent, remember how catabolite repression, so this one also involves catabolite repression mechanism. If glucose is absent, the presence of CRP bound to the site of AraO1 helps break the DNA loop and helps AraC to bind the AraI. Now you have this, see the difference between, when there's absent, it becomes like this. If it's Arabinose is present, then it becomes like this, the arrow, right? So then when you have this, you get positive activation of the promoter. So again, then you may want to have a look at this picture. It's the same thing. Without AraC, sorry, without Arabinose is that black small thing. Without Arabinose, AraC binds AraO2 and AraI, makes it a loop. It also binds here to prevent production of AraC. But in the presence of Arabinose, the loop is broken. Now, instead of preventing transcription activity, now it promotes transcription. And then that happens when you have CAMP at cap site. Right, and this is the same thing, almost the same mechanism that you see in like operon. When glucose is absent, you get that binding. So both when you have Arabinose as well as absence of glucose, then you get transcription. All right. So in conclusion, the Arabinose operon demonstrate both negative and positive control by the same protein. So that's why it's an example. All right, so remember, just recap again, you have your like operon, which is controlled by the repressor molecule, also by catabolite repression. You have the Tryptophan that is controlled by the repressor molecule and also by attenuation. And then you have this system where AraC can either, depending on the presence and absence of Arabinose and also absence and presence of glucose, it has different conformation, allosteric conformation. It has different properties. It can either bind and produces a loop, or it can break a loop and then binds to promote switching on the Arabinose operon. All right. Okay, so I think that marks the end of your operon lectures.